Plant breeding plays a vital role in meeting the food demand of the increasing world population. While more than 840 million people around the world lack daily meals, it was reported that more than 3 billion people are fighting hidden hunger. Hidden hunger refers to a poor diet or malnutrition, that is, a deficiency of essential micronutrients, proteins, and vitamins. As poor people cannot afford fruit and vegetables that are rich in nutrients, they survive on a diet containing staple food crops such as wheat, rice, and maize. However, the level of vitamin A and micronutrients such as iron and zinc is very low in such crops, which makes them vulnerable to deficiency diseases that affect physical and mental health. To overcome this problem and promote public health, initiatives like biofortification were introduced. Biofortification is selective breeding of crops with a higher nutritional value such as proteins, vitamins, and minerals. For example, Atlas 66, a hybrid variety of wheat rich in protein, is bred with native wheat crops to fortify them. As a result, biofortification results in high yielding crops fortified with specific nutrients. Another example of a biofortified staple crop is an iron fortified rice variety that is 5 times richer in iron compared to its native variety. Similarly, maize hybrids containing double the quantity of amino acids compared to the other hybrids was developed. Did you know that the Indian Agricultural Research Institute, New Delhi, used biofortification to develop several fortified fruits and vegetables? Examples include vitamin A fortified carrot, spinach, and pumpkin, vitamin C fortified tomatoes, mustard, and bitter gourd, protein fortified beans and cotton peas, and iron and calcium fortified spinach. Despite biofortification and several plant breeding techniques to boost food production, the demand for food was ever increasing. The shifts to meat diet further increased the demand for grains, as food grains are also used as animal feed. For example, a cow needs to be fed around 7 to 10 kilograms of corn to get around 1 kilogram of meat from it. For this reason, in addition to biofortified foods, scientists proposed the use of microbes as a food and feed supplement. These single-celled microbes are rich in proteins and are referred to as single-cell proteins or SCP. And so, they are cultured to be used as a rich source of protein food. For example, spirulina, a blue-green algae, is cultured for its high protein and mineral content, and it is also a source of vitamins, carbohydrates, and fats. It is used as a food supplement for human beings and as a feed supplement for animals. Moreover, it helps reduce environmental pollution as it can grow on wastewater from potato processing plants, straw, molasses, animal waste, and sewage. Another example is Methylophilus methylotrophus, the bacterium that can produce 25 times of proteins. Nowadays, fungi such as mushroom are accepted as edible. Scientists believe that soon microbes too will be accepted as a food supplement. Tissue culture is a method used for plant propagation in addition to other traditional plant breeding techniques. It was during the 1950s that scientists found that any part of the plant grown in nutrient medium under sterile conditions can give rise to a whole new plant. This property of plant cell to give rise to a whole new plant is called totipotency. Tissue culture uses this property of plant cells for asexual plant propagation in an aseptic environment. It requires a nutrient medium that contains sucrose, inorganic salts, vitamins, proteins, and growth regulators such as auxins and cytokinins. The tissue culture technique involves several steps. The first step is excising a small part of plant tissue like the root, shoot, or leaves of a plant. Then placing the cutout part or explants in a suitable nutrient medium, followed by allowing the explants to multiply and form a group of tissues called callus. The callus is then allowed to multiply to form many plantlets, which are then transferred to a different container for rapid multiplication. Finally, the developed plantlets are transplanted to the soil for normal growth. Therefore, tissue culture is performed using a small part of a plant to develop many plants, and so is also called micropropagation. As all these plants are developed from the same part of the parent plant, they are genetically identical and called somoclones. Micropropagation or tissue culture is employed in the large scale production of tomato, banana, and apple. This technique can also be used to propagate high yielding disease resistant varieties in a short span. Even if a plant is diseased, tissue culture can be used to get a healthy plant using the explants of apical or axillary meristems. This is because the meristem is always free of disease and microbes. Culturing of meristems can be successfully carried out in plants such as banana, sugarcane, and potato. In addition to asexual propagation of plants, tissue culture is also used to make two different species of plants that are otherwise sexually incompatible. Here, individual cells are isolated from each plant species and are placed in a culture medium. The cell wall gets digested in both cells, which aids the fusion of two protoplasts of different species, resulting in a hybrid protoplast. The hybrid protoplast is called the somatic protoplast, which gives rise to a whole new plant and may contain the desired characters of both species. This process of developing a hybrid plant using somatic cell is called somatic hybridization. It was employed while using the tomato and potato plants to obtain a hybrid plant. As a result, the hybrid plant resembles the tomato, but it did not combine all the desired characters as expected. In this manner, tissue culture, somatic hybridization, and other plant breeding techniques provide a huge potential for manipulation of plants to produce new varieties. boost food production, and enhance crops.